So we're looking at chapter 6.3, Defense Against Infectious Disease. An important distinction that we need to make right here is that we are looking at infectious, meaning that it is coming from something communicable, something that can be shared, and therefore something which is caused by a pathogen. And we'll look at exactly what that definition is here in just a second, but we are not talking about non-infectious diseases. Anything that cannot be transmitted, for example, cancers or poisons, etc., are non-infectious because you're not passing them from one person to another. I'm going to put a little asterisk here next to HPV. So we're only looking at infectious disease. So to make sense of the chapter, we first need to look at a couple of definitions: pathogen, antigen, and antibody. We'll start off by looking at what is a pathogen. This is any virus, bacteria, fungi, etc. And as a reminder, with the exception of viruses, all of these things are living. And we know that living organisms have cells, and that all cells have a cell membrane. And that on the cell membrane, there are all kinds of different proteins, right? And they can differ. There can be a whole bunch of different types. Okay? And so each one of these proteins, which has the ability to cause an immune response, each one of these is called an antigen. So your body hosts an immune response and makes antibodies, which are actually much, much smaller than this, in response to the antigen. So that would look something like this, just tiny little proteins that fit together. And for every antibody, there's one antigen. So they are very specific to each other. The objective 6.3.2 asks you to explain why antibiotics are effective against bacteria, but not against viruses. We'll start by taking a look at how do antibiotics work. There's two ways that they're primarily going to work, either by stopping transcription and translation, or by breaking the cell membrane. Now, both of these techniques are going to be great at stopping them from reproducing, right? Puncturing some holes in the cell wall or the cell membrane of the bacteria, or stopping transcription and translation from happening. And both of them rely on the fact that cells are living organisms and therefore have characteristics of life, such as a cell membrane and its own metabolism. And remember, this chemical reaction of making more DNA is metabolism. We don't tend to think of it as such very often, but any chemical reactions that are happening within an organism is a metabolism. And bacteria, which are prokaryotes, do have metabolism. So now we move on to the second question, which is how do viruses infect? And in fact, they are non-living organisms. They have like a weird hybrid. Who knows if they're living or not living, but regardless, they either have DNA or RNA, single or double-stranded, depends on the virus, trapped inside of a capsid, and a capsid is a protein coat, and they're going to come on to a cell, either a prokaryote or a eukaryote. And in the case of a pathogen that would infect us, we'd be thinking this would be a eukaryotic cell right here, and they're going to inject their DNA or RNA and commandeer all of the cell's machinery. So any ribosomes are then going to start to manufacture the proteins to make more little viruses. And we're really thinking tiny, 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 not nearly as big as I've drawn them here. But thousands and hundreds of thousands more viruses are made. It looks like a cat. That's all right. That causes the eukaryotic cell or the host cell in this case to pop open, spill out the contents of all the viruses, and allowing the continue to keep going. And finally, we need to take a look at why don't antibiotics work against viruses? Sweet and simple, because viruses have no metabolism. They rely on us. Without us, there is no life in them. So we can take advantage of the fact that bacteria have their own metabolism, which is so different than ours, and their structures, which are so different from ours, and be able to find ways to attack bacteria. But we cannot find a way to attack the virus's metabolism because, well, they don't have one. They're commandeering us. And of course, antibiotic resistant. We'll keep this short and sweet. We have a bacteria, and it divides a whole bunch of times, and they divide a whole bunch of times, and so on and so forth. And eventually what you're going to end up with, someone somewhere is going to have a slight mutation. And maybe in a normal situation, they would have a lower chance at surviving. But if you have an antibiotic which is able to wipe out the rest of the population, and this little guy has some kind of benefit conferred, which would be resistance to a particular antibiotic, he's going to be able to continue to divide and make a new population within you that is now resistant to whatever the antibiotic that you started out with. Penicillin, streptomycin, anything like that, all of them have bacteria which are resistant to them, which means we're putting ourselves at a pretty constant risk by taking antibiotics unnecessarily. So only take these as prescribed. Take them for the full course, 10 days typically. Don't skip a dose. Don't stop using it once you feel better. You need to continue your entire dose, and don't ask doctors for antibiotics. Don't take them for flu. It's not going to help. So there we go. So we're going to start talking about the skin and mucous membranes. These are your first line of defense against any kind of invaders that are trying to kill you. We'll start by talking about the skin and how it's important. So here we go. Skin. Why is it important? It's a continuous covering over your entire body, and that's going to make it really hard for anything to find entry into your body, with the exception of where you have mucous membranes. And it's why when you have a cut, you want to be careful about getting it bandaged up quickly so that bacteria can't get in. It's a pretty thick and tough barrier, especially against the size of different bacteria. So that's not saying it's completely impenetrable, but things that do get onto it, especially bacteria, tend to not be able to get very deep. They can get to the epidermis. That's the outer layer. And then you've got the dermis immediately underneath. The dermis is living. It's creating new skin and pushing it out. Once you get to the epidermis, it is all dead cells. So this is constantly flaking off, making it even harder. It's kind of like climbing up a waterfall. Your skin is also fairly dry. There's something to be said about a warm, moist environment. Yeah, I said that. Moist. Anyway, a nice moist environment is going to be fabulous for fungi and bacteria to grow. If it's too dry and too salty, it makes it really difficult to grow. And lactic acid is always being excreted in small quantities, dropping the pH of your skin to between about 5 or 6, which is lower than what would be favorable. And lysozyme is the enzyme which is secreted onto your skin that's going to help to break down different pathogens that try to attach to you. And lastly, you are covered in bacteria and fungi and weird little microbes that you don't even want to know about. But what are they doing? Well, they're acting as competition. So anybody else that tries to grow in that area ain't going to work. Well, hopefully not. So by having all of these bacteria in your gut and on your skin, these are working for your advantage. They're not trying to hurt you. They're just enjoying the nice, cozy conditions that you're providing to them where they're able to. So they're stopping anything else from being able to grow. And next we need to look at the mucous membrane. So anywhere you don't have skin, you have mucosal membranes. And why is that important? Well, what are they doing? They're secreting mucus. And mucus is going to be really sticky, so anything that tries to get in is going to be trapped. They also have lysozymes in the mucus. And an unfavorable pH. So where are you going to find mucosal membranes? Well, anywhere you have a natural break in the skin that needs to be there to gain entry for other things into the inside of your body, namely your mouth, because you need a way to get into the esophagus so that you can eat and digest. The nasal cavity, you need to breathe. I hope that's pretty self-apparent. And the nether regions, for obvious reasons, being able to excrete waste. And the vaginal and penile regions. Yeah, you just got me on video saying vaginal and penile regions as well. And for this objective, you need to know about 6.3.4, which is how phagocytic leukocytes can ingest pathogens in the blood and in the body tissues. So a quick recap. When we talk about blood, there's 4 main things that are in the blood. Starting from the bottom, we've got the plasma, which is carrying most of the nutrients, hormones, antibodies, just the liquid portion that's carrying the majority of non-cellular material. You've got platelets, which are responsible for clotting. Skipping up here, RBC, or red blood cell erythrocytes. Those are carrying hemoglobin, which is carrying oxygen. So the RBCs are responsible for transporting oxygen. And then your white blood cells, your WBCs, or your leukocytes. One type of leukocyte is a phagocytic leukocyte. And all of your white blood cells mostly have to do with your immune system. The phagocytic leukocyte, as the name would imply, are phagocytic, meaning they do phagocytosis, the form of endocytosis, bringing invaders into the body for their destruction. So to start this little adventure off, we need to start with a little phagocyte, and he's going to see a little bacterium and go after it using the process of chemotaxis, meaning moving towards the chemical substance which is being released. Chemo for chemical, taxis, moving towards, right? You take a taxi, you get this concept. And when the bacteria gets close enough, actually when the leukocyte has chased the bacteria far enough, it'll form a small invagination. And that's going to be something in which kind of wraps around this little bacterium or virus, or even some larger things such as protozoans, until finally it has it completely ingested in its own little vesicle. And this vesicle is going to merge with another vesicle, and this one's going to be filled with lysozyme, meaning that this vesicle is a lysosome. Lysozyme is the enzyme. Lysosome is the organelle, as we'll call it. And these two vesicles are going to merge together. And that is going to cover the little bacteria friend here in lysozyme until finally we are able to break him down, as it should be. So we're going to pick up right where we left off, but start talking about antibody production. Okay, so we've got our phagocytic leukocyte. He's just ingested bacterium using phagocytosis. So here's our little phagocytic friend. And we've got the bacteria in there, and the lysozyme merged, or the lysosome merged in, so that lysozyme begins to cover the bacteria, and it starts to break him down. But this is not going to break down the bacteria or the invader, the pathogen, completely. Instead, what it's going to do is leave kind of some chunks. Those chunks are going to be important. And those chunks are then going to be transported by the phagocytic leukocyte onto the surface of our cell. Okay, so this is called presenting. The process of presenting is going to take this little guy here. Now, this was on the pathogen, and because it's a protein that causes a response, so a protein that causes antibody production. This is called an antigen. Remember, pathogens have antigens, and they're proteins that cause antibody production. And the way that they're going to produce the antibody is that this little antigen is going to trigger the activation of a T cell. And that T cell has a special receptor that says, yep, you're the right guy that I'm looking for, okay? So the T cell then becomes activated, and he gets all excited, and in turn activates a different cell called a B cell. Both of these, the T and the B cell, are types of lymphocytes. Okay, so the B and the T cell have now worked together collaboratively to turn an antigen, so turn a crisis situation into a situation where they can start going on the offense. T cell, remember we also talked in class that it's called a T helper cell, which is just one type of T cell. But specifically here in this cascade, we're looking at a helper. So let me switch over to, here we go. So this right here is our phagocyte. the phagocytosis, whatever. There's a verb for that, but the bacteria is going to be broken down until all that's left are little antigens, the little proteins that were on the outside. That gets presented. The presentation process then activates a T helper cell or a T cell. That T cell in turn is going to then activate the B cell. And B, we remember B for body because they're the ones making antibody. So antigens made by the pathogen, antibodies made by your body, by the B cells. And now the antibodies are going to start being produced by the B cells. And they look like little Ys. And they can be either excreted, so they'll kind of grab them. Well, that didn't work. But they can either grab these little antibodies and send them out of the cell so that they start free-floating all throughout the rest of the body in the plasma. Or it'll attach it to the outside of this cell, so that as an entire cell, it can find other antigens. So how do antibodies actually work? Well, they've got two different regions. All of this down here is always the same. The only part that changes is the variable region. The variable region is called variable region because it is variable. So maybe it's looking for something like this. And so it's just going to happen to find an antigen that has exactly the same shape. So maybe this is the cell of a bacterium, and this is much more in proportion to what we've been seeing into class. So this bacterium would be huge as compared to this fairly small little protein of an antibody, also abbreviated AB. And so when this antibody catches on to this perfectly fitting little cell down here, there's a match and they grow in together. And again, I can't move things like I'm wanting to, but it'll kind of move itself up here and latch on, kind of like an enzyme does to a substrate. They fit perfectly, they latch on together, and then the antibody can go either to attack the bacterium here or can at least neutralize it. We'll talk about that later in higher level.